During cellular respiration, a glucose molecule is gradually broken down into carbon dioxide and water. Along the way, some ATP is produced directly in the reactions that transform glucose. Much more ATP, however, is produced later in a process called oxidative phosphorylation. Oxidative phosphorylation is powered by the movement of electrons through the electron transport chain, a series of proteins embedded in the inner membrane of the mitochondria. Let's start by looking at cellular respiration at a high level, walking through the four major stages and tracing how they connect up to one another. Glycolysis is the first of the main metabolic pathways of cellular respiration to produce energy in the form of ATP. Through two different phases, the 6-carbon ring of glucose is cleaved into two different carbon sugars of pyruvate through a series of enzymatic reactions. The first phase of glycolysis requires energy, while the second phase completes the conversion to pyruvate and produces ATP and NADH for the cell to use for energy. Overall, the process of glycolysis produces a net gain of 2 pyruvate molecules, 2 ATP molecules, and 2 NADH molecules for the cell to use for energy. In the first half of glycolysis, 2 adenosine triphosphate ATP molecules are used in the phosphorylation of glucose, which is then split into 2 3-carbon molecules. In the second half of glycolysis, energy is released in the form of 4 ATP molecules and 2 NADH molecules. So far, glycolysis has caused the cell 2 ATP molecules and produced 2 small, 3-carbon sugar molecules. Both of these molecules will proceed through the second half of the pathway where sufficient energy will be extracted to pay back the 2 ATP molecules used as an initial investment while also producing a profit for the cell of 2 additional ATP molecules and 2 even higher energy NADH molecules.
Anaerobic respiration. How cells extract energy from glucose without oxygen. In gist, the anaerobic reactions make alcohol, while in your muscles they make lactic acid. In lactic acid fermentation, NADH transfers its electricity directly to the pyruvate, generating lactate as a byproduct. Lactate, which is just a deprotonated form of lactic acid, gives the process its name. The bacteria that make yogurt carry out lactic acid fermentation, as do the red blood cells in your body, which don't have mitochondria and thus can't perform cellular respiration. Fermentation is another anaerobic or non-oxygenic requiring pathway for breaking down glucose, one that's performed by many types of organisms and cells. In fermentation, the only energy extraction pathway is glycolysis, with 1 or 2 extra reactions tracked on in the end to regenerate the electron carrier NAD+ from the NADH produced in glycolysis. 2. Pyruvate oxidation. If oxygen is available, aerobic respiration will go forward. In eukaryotic cells, the pyruvate molecules produced at the end of glycolysis are transported into mitochondria, which are the sites of the cellular respiration. There, pyruvate will be transformed into an acetyl group that will be picked up and activated by a carrier compound called coenzyme A, CoA. The resulting compound is called acetyl-CoA. Acetyl-CoA can be used in a variety of ways by the cell, but its major function is to deliver the acetyl group derived from pyruvate to the next stage of the pathway in glucose catabolism. Step 3. Following glycolysis, the mechanism of cellular respiration involves another multi-step process, the Krebs cycle, which is also called the citric acid cycle or the tricarboxylic acid cycle. The Krebs cycle uses 2 molecules of pyruvic acid forming glycolysis and yield high-energy molecules of NADH and FADH2, as well as some ATP. The Krebs cycle occurs in the mitochondria of a cell. We arrive to a final step of cellular respiration, oxidative phosphorylation. The electron transport chain is a series of proteins and organic molecules found in the inner membrane of the mitochondria. Electrons are passed from one protein of the transport chain to another in a series of redox reactions. Energy released in these reactions is captured as a proton gradient, which is then used to make ATP in a process called chemiosmosis. Together, the electron transport chain and chemiosmosis make up oxidative phosphorylation. And with that, we conclude with the whole process of cellular respiration, obtaining up to 38 ATP molecules for every glucose.